Hello, team IBBC31.
Yes.
Turkey, correct? Okay. You have 3 students in your team, right?
Yes.
Student. Okay, thank you. And welcome to the Global Youth Science and Technology Bowl 2020. Your team, your project is about effects of Propolis and Curcuma Longa on cancer, right?
Yes.
Right.
Yes. Okay. Thank you so much for attending the online judging today and for your effort made in the past few months. Today, we are happy to have Professor Lam Yunhua from City University of Hong Kong, Dr. Kathy Lui from the Chinese University of Hong Kong, and Professor Ai Sai Ming from Chinese University of Hong Kong to be the judges. Okay, thank you. Okay, you will have 5 minutes to present your projects. When time is up, we will stop your presentation. After that, there will be a 4-minute Q&A. You will be able to see a timer on my screen. So you may share your screen now, team.
Okay.
Thank you.
Can you see it?
Yes.
Okay.
How about judges? Can you see their PowerPoint sharing? Okay. Okay. Are you ready to start now?
Yes.
Okay, please start.
Hi, I'm Selin Aşık.
I'm Arda Deniz Karaman.
I am Arda Yıldırım. We are from Turkey, Istanbul, Okyanus Colleges.
Title of our project, an investigation on the effects of Propolis and Curcuma longa on cancer, observing their impacts on vascular formation and chicken embryos growth. At the beginning of our presentation, I want to tell you what is cancer. Cancer is a group of malice diseases which appear as a result of degeneration and uncontrollably propagation of cells. As a result of improvements in life expectancy in last 100 years, the discrepancies and anarchy in the reproduction of cells in organs and tissues and complicated growing have made cancer common among the people. Cancer comes second after cardiovascular illnesses and human mortalities and appears in 1 out of 250, 300 in Western societies. 1 in 300 people over 60 years of age. That's why the diagnosis and treatment of this illness is very important. In this project, our objective is to find out which Curcuma longa and Propolis dosages are more effective on vascular formation in egg embryos. That is, this study discusses the preventive impacts of Curcuma longa and Propolis on vascularity in egg embryos. Cancer cells spread fast and metastasize, forming their own blood vessels. Their effects on cancer will be studied by observing which dosages of these substances to be used are more effective on vascularity to what extent.
Curcuma longa plant is known to have been used in Indian medicine for a long time in the treatment of flu, cough, liver disorder, rheumatism, sinusite, and onyxuria. Curcuma longa used as food colorant, is colorless, heat resistant, and composed of tetrahydrocurcumin, which is antioxidant. Propolis became known in the treatment of disease 1,000 years ago, and it was used by Egyptians, Greeks, Romans as a natural remedy either in the cure of many illnesses or palliative purposes.
And the development process of chick embryos is very complicated and spans around 21 days. In this process, there are 2 states, and external interventions such as medication and injection application should not coincide for the 2nd hour and 20th day of their growth. And their cardiovascular systems start to form at 25th hour. Angiogenesis is the process of the formation of blood vessels, and it can be controlled with chemical substances, some of which repair the damaged blood vessels and stimulate the formation of new ones. And angiogenesis is also crucial in the growth and spread of tumors, since the formation of new blood vessels help nutrients reach to cancer cells. Thus, growing cancer cells start to spread into adjacent tissues and other organs through the body. Tumors cannot grow and develop without blood vessels. Hence, the substances preventing the formation of blood vessels have been the subject of scientific studies.
At the beginning, we prepared Curcuma longa and Propolis dosages. We prepared 4 different dosages from each substance. From Curcuma longa, as you can see, 20, 40, 60, and 100 milliliter were used. And from Propolis, the dosages of 0.1, 0.2, 0.3, and 0.5 milliliter were used. To extract the curcumin active substance from Curcuma longa and to dissolve Curcuma longa, 10 milliliter ethanol and 240 milliliter distilled water were mixed to get 250 milliliter mixture. Then this mixture was sterilized in autoclave and this mixture was diluted with distilled water. 50-gram curcuma longa was added into this solution, obtained to get extract substance released into the mixture. It was left to cool at room temperature. After the cooling process, it became ready to use. The eggs to be used had embryos and they were newly fertilized and they were used on the same day. The extract acquired was injected into the eggs and switch crack covered with wax. After being kept in incubator for 96 hours, vascularity index was tested. The experiments were repeated 3 times and analyzed whether the results were coherent or not.
At the end of the 96 hours, the eggs taken from incubator were broken and embryo development and vascularity were examined. It has been found that in correlation with the increase of curcumin dosage, the vascularity formation in embryo decreased. In control group, in the eggs into which 1 milliliter distilled water was injected, there were heartbeats at the end of the 96-hour growth. As to experiment group, the vascularity decreased in parallel with curcumin dosage increase. As to Propolis experiment, it found that at the beginning, the vascularity decreased in parallel with curcumin dosage increase. But later, it was observed that Propolis had a toxic effect on the embryo. In the Propolis dosages of, as you can see,
Okay, team,
0.5.
time's up for a presentation. Thank you so much. Now